All right, questions. So we will now talk about humoral immunity, process of clonal selection, roles of plasma cells and memory cells in humoral immunity, and active passive humoral immunity, and then antibodies. Right? So what is humoral immune response? Firstly, which cell is responsible for humoral response? Is it T cell or B cell? B cell. So what happens here when the B cell now first encounters this target antigen, which is in the lymph node, again, that will provoke humoral immune response. Okay. So first time they are getting now activated. So antibodies specific for that particular antigen will be then produced. So firstly, they will have to encounter the antigen. They're activated, the B cells, and then they will start producing the antibody. So that's the story, basically. First, they have to encounter, and then they will produce. So B cells are activated when antigens bind to them, like cross-link them. And then this triggers the receptor-mediated responses. And then they create effector cells, and then clonal cells are produced. Okay. So we'll come back to this. So this is the whole process of. So there is a primary response for B cell for when clonal selection is happening, and then there is a secondary response. So what happens? Firstly, there are B cells. So they are now in the lymph node, suppose, but they're not yet active. So in the first step, they encounter the antigens. They encounter the antigens like you see here. Now they are encounter means now they're activated. So the next step is getting activated B cells, right? So they are now activated. Once they are activated, what they will do, they will start proliferating to form a clone. Clone is simply a copy. Okay. Clone is a copy. Now, once they make one copy, now what happens to this clone? These clones actually make two kinds of cells. The clone itself one of them will make a memory B cell, and the other will become plasma cell or effector B cell. So two clone cell, each will divide to form two effector cells or plasma cells, and the other clone will also produce one plasma cell, but it will also produce a memory B cell. So in turn, basically from two clone cell, three plasma cells or effector B cells are produced, and one memory B cells is produced. So this is the primary response. And what is the job of this plasma cell or effector B cell? This is the one that creates the effect, means it produces the antibodies, really, right? So they are the one that are producing the antibodies. The effector B cell, that is also true for T cell. The effector cell is the one that creates the effect. Okay. So here the effect is producing the antibody is the effect. Now, does the memory cell do that? They don't. They are memory cells. So they will remember this whole thing. Like what kind of antigen they encountered, they will remember that. Now, if there is a subsequent similar challenge being produced to the body, like say encountered that antigen one more time, then this memory B cell will remember that. And what they will do, this can be like 5 years later. You had a disease today, and 5 years later, you're having that. These memory cells will be in your body, memory B cells. So after then, they will again start now proliferating. So they are already immunocompetent. They have gone past through self-tolerance, immunocompetent, active, everything. They still remember what they did 5 years before. Now here, they will start proliferating and produce more plasma cells. Okay. And those plasma cells will again produce more of the same antibody. And they will also make sure to produce some more memory B cells. Okay. So then these memory B cell will do the same thing if this again happens to you maybe 15 years later. Okay. You should have some kind of defense. So therefore, clone cell that do not become the plasma cell, they become the memory cell. Okay. And what do this memory do? They provide us immunological memory. Means are simply means that they mount an immediate response to future exposure to same antigen. Something happened today, 5 years later, you encounter the same antigen, it will remember what to do, those memory cells. You encounter that 15 years later again, will have some form of defense against that. So this is called as clonal selection of a B cell. Okay, so they do have memory, and then the effector cell is the plasma cell. So the memory cell do not produce the antibody. Then which cell produces? Which one? The plasma cell is the one that produces the antibody. The job of the memory cell is just to remember what it's supposed to do. Okay. Now, when it comes to immunological memory, two things can happen. So firstly, suppose there is like an antigen. So the antibody A is produced. Initially, if you get an initial exposure, something brand new, it will take 3 to 6 days to trigger or to produce, to undergo all these processes, and then finally start producing the antibody. So you have a brand new infection, it will take our body to develop, go through all the clonal selection and produce the antibody for a couple of days. It can vary from person to person, typically 3 to 6 days. Okay. Now then it will reach the peak around 10 days. Okay. And then it will decline. So after 10, 8, 9, 10 days, this will decline. Now by this time, your infection also will be reduced, but the body will retain what kind of cells? The memory cells, yes. So the memory cells will be there. Now you have a re-exposure again, couple of years later, couple of months later, days later, whatever. Then these memory cells will have this immunological memory. They already have gone through all that clonal selection. So this time the response will be much more faster. It will not take 3 to 6 days. It could be within hours after you got exposure. Okay, so response within hours, not days. And then the antibody level will peak within few days at much higher level because it already knows what to do. That's why if you see here the plasma cells in the secondary response are much more produced. So the amount of antibody produced for secondary response is way more than the primary time, the first time. Okay. So in this case, the antibody will now bind with greater affinity. It will know what exactly to do. It will stay there. Like you could see, there's a stable phase. So it will stay there for quite some time, weeks to months, and then gradually it will decline. If you have a second antigen, antigen B will have the same response of antibody B, will do the same thing. So that is the difference. Primary immune response takes a little bit of lag, means it is like lagging behind, right? So it will take a little bit lag time to trigger, but if it triggers, then it will give you the protection, but then it will decline also after a couple of days. But if you're re-exposed, then it does not have to go through that lag phase because it already knows what to do. So it immediately will trigger and will stay in your body for quite some time. And basically, it will heal you much more faster if you already had the disease. All right. Now, active and passive humoral immunity. What does this mean? Humoral immunity, this is done obviously by the B cell. Now, this occurs when the B cell encounters specific antibodies and we acquire immunity. This can be active or passive. What is active? Active also has two subtypes. One is naturally acquired, one is artificially acquired. You are first time getting infected by a virus or a bacteria, you naturally will acquire an immunity. The response thing, the whole process is the same thing that we just discussed, like we spoke about, the clonal selection, producing memory and effector cells. But you are first time getting exposed to something really new. That is naturally acquired immunity against infection of virus or bacteria. And that is active because it is producing an active response. You are getting it directly. Now, but if you are given a vaccine or a dead pathogen, same thing, then you are also you are getting an active response, but you're not getting it naturally, you are getting it artificially. Okay. So body will still develop those memory cells will still develop those plasma cells, but in this case, it will be done artificially through the vaccine. All right, so that's called the active immunity. Now, what is passive immunity? We also develop passive immunity when some ready-made antibodies are given to you, but you are not directly getting it. For example, the best example is when an infant gets antibody from mother's milk. That's why it is said that mother's milk is very important for an infant, because that's rich in antibody, right? It helps the infant. So therefore, they are passively getting it. They are still naturally getting it from the mother, but not directly. Again, it's a passive system through another person. So that's why it is naturally acquired, but a passive system. Okay, it's a passive system. And a passive system also can happen artificially. For example, if you're given a serum itself, so you are given an injection with antibodies directly. That is a passive system, but artificially given. There's a difference between vaccine and artificially acquired injection, because when you're directly given with the antibodies, you're directly giving the antibodies. But if you are given the vaccine, then you will still have to go through the process of the clonal selection. Does it make sense? Because when you're just given the vaccine, you're just giving the pathogen again. It's a dead pathogen, but still pathogen. So you'll have to still, that's why you get fever when you're given a vaccine. But if you're given a medicine, you may not go through the fever, because it is already the ready-made antibody is already given to you. But if you're a vaccine, it's a dead pathogen, but still a pathogen. So you will go through the clonal selection process and you will develop your own memory cells, own antibodies. But that is the difference. And same thing for mother's milk or placental connection. You are given ready. It is a natural way. It's not someone is injecting. It's a natural way, but you are given the ready-made antibodies. The infant's body is not producing it. Make sense, everyone? Now, what are these antibodies? These are called as immunoglobulins or IgGs. These are proteins secreted by the plasma cells, of course, and they're made up of gamma globulin, capable of binding specifically to antigen. Obviously, that is detected by B cells. There are 5 classes: IgM, A, D, G, E. MAD-GE, actually, MADGE. All right. Questions? So we will have to know about each of these categories one by one. Firstly, the basic shape of antibodies like this. It looks like a Y, okay? T or Y. There are 4 polypeptide chains. So this is a protein again. So they will have polypeptide chains. So there will be 4 chains generally. 1, 2, 3, 4. 4 polypeptide chains. Okay. 1, 2, 3, and 4. And now 2 of them will be the heavy chain. Like this, you can see the inside ones are the heavy chains. They're the bigger ones. And the outside ones are the lighter chains. These are the smaller ones, L chains. Okay. Light chain, heavy chain. Now, how are they linked with each other? They're linked with this disulfide bond. Like any other polypeptide, they will be linked with this disulfide bond. All these chains will be linked together by the disulfide bond. Now, the region that is in the open area that actually encounters with the antigen is called the variable region. So, which is this particular section, you can say this is the variable region, because this is the region where they are encountering, this is the variable region where they are encountering the antigen. But the inner section, this inner section, this is the C region, which is here, this whole section, this is the C region, constant region. So the constant region, of course, directly do not encounter the antigen, but it determines the class of the antibody by the structure. Okay, so this is the effector region, determines what kind of antibody it is, but the variable region is the one that encounters the antigen. That is common for all antibodies, the basic structure. Now, how are they different? Firstly, the M. M is the first antigen that is produced by plasma cell for primary response. Okay. First time you encountering an antigen, your body will produce M antibodies. All right. They're big in structure. There are 5 monomers. 1, 2, 3, 4, 5. Each of these are monomers. So each of them, these are all monomers, and each of them have 4 polypeptide chains. So it's a pentamer structure, actually. 5 monomers joined together. What do? They readily activate the complement system. Like I told you in the first class, that adaptive and innate immunity, they work together. So antibody itself will not be able to destroy the antigen. It will mark it, but the actual stuff will be done by complement or phagocytosis, something like that. Okay, so here IgM will take help of the complement system itself. Okay, once it is activated, and it circulates in the blood plasma and has numerous antigen binding sites. The next one is IgA. This is mostly a dimer. Sometimes dimer means there will be 2 monomers present. Sometimes could be a monomer 2. And it helps prevent the entry of pathogen. Okay. And this is slightly less present in plasma. It is mostly present in the secretory fluids like sweat, intestinal fluid, saliva. So something is trying to enter in your body, then sweat, saliva will try to prevent it, because sweat, saliva also will have these IgA antibodies. So that's why it is called as it's a secretory antibody. Okay, so anything that is secreted from your body might have these things. The third one is IgD. Okay, this is a monomer. So the next 3 ones, D, G, and E, these are the smaller ones. They are mostly monomers. The D, for example, is only found on B cell surface, and they function as a B cell antigen receptor. IgG, this is the most abundant antibody. The 75 to 80% of antibodies in plasma is actually IgG. It can form a primary response as well as secondary response. Okay. In the so whenever we are triggering primary response, we can either we will produce IgM, but also we can produce IgG. But for the secondary response, we produce a lot of IgGs. Okay, so this also works in a very similar way, like the M works, activates the complement, activates the complement system, protects against virus, bacteria, and that. Also, this has a unique ability of crossing the placenta, okay, and conferring passive immunity from the mother to the fetus. So fetus also needs protection. So IgG is the only one that does it best, like successfully, actually crossing the placental barrier. And E is also the last one. This one actually is a small one, and it gives us protection against allergies and parasitic infections, causes mast cells and basophils to reduce histamine. Okay, so if you have anti-allergic, anti-parasitic infection, then specifically we will produce E. So these are the 5 classes. Now, there is a unique property of antibody that they can switch classes among them. So plasma B cells can, for example, IgM and IgG, like I just said, IgM and IgG, they switch all the time. So you have a primary response, we will produce IgM, but we can also produce IgG, and they can also switch in between. So IgM and IgG, it is very, like, it's not a very abnormal thing to switch between the classes. But the others also can switch a little bit also. Now, antibody targets and functions. This is the critical thing. Antibody do not destroy antigen. What do? Like I told you before, they inactivate and tag them, and then they produce the antigen antibody complex. Antigen antibody complex. Who destroys these complexes? The innate system. Give me one example or 2 examples. Macrophages, complement, phagocytosis. So those are the actual process that can destroy them. But what antibody does is specifically identify them, tags them, forms the complex, and it determines the pathway. But actual work is done, could be done by these innate pathways. Okay, the next actually is this case study that you actually have as your assignment. It's actually part of this for assignment. So Max, this case study says Max, a person, is bitten by a rattlesnake while on a camping trip. His friends actually, when they detect, they immediately apply ice packs to the bitten area to slow the spread of the protein-based toxin and then rush him into emergency. What kind of treatment do you think will be given to him and why? So I'll give you some clue. I'm not going to tell you the answer right now. So basically, he is already being presented with a pathogen that was not there before. But this is a poison, okay? So it's not a virus, it's a poison. So he needs to have a faster response against the poison. Otherwise, he might die. So think about, that's your clue. So think about it and we will talk, we'll pick up from here on Wednesday. So remember, this is a poison. So that's the key.